basically in the law of dominance, the allele which suppresses the other allele expression. Okay, when I am trying to talk something, when I'm trying to talk something, one of my brother is not allowing me to talk, so that my brother is a dominant allele for me. In this context, I'm a recessive allele. I can't express, I can't bring any character. Okay, so that law, that concept explaining law is called law of dominance, right? So now, one by one, we'll discuss in detail. You not to worry about it, right? Before going to this law of dominance, I explained about what is my dominant allele, what is my recessive allele. Now, before entering into dominant allele, recessive allele concept in depth, can we give the example for the dominant allele? Can we give the example for the recessive allele? Yes, of course. Okay, the tall is a tall character is controlled by dominant allele. Okay, dwarf character is controlled by recessive allele. Okay, so like this, what we need to give the contrasting character is fitting or fixing in which particular domain, dominant or recessive, right? So now, if you're entering into the monohybrid cross, it's a cross made between the two individual varying in one character. Okay, the two plant varying in only one character. Okay, and those two plants we taken for crossing. Okay, how we do crossing? By transferring the pollen grain from one plant to other plant. What do you call that? Pollination. Because of pollination, what happens? Fertilization happens. Because of fertilization, what happens? New progeny comes. So this is the way you need to study. Okay, here there are some students. Okay, here monohybrid cross is a cross which is nothing but the alleles are responsible. Don't have wavering mind. Study with happiness. Study with joy and cheer. Right. So in the monohybrid cross, when we are doing the cross between the two individual varying in one character, we'll be getting the one particular hybrid, okay, or two particular hybrid you are getting. Like this, we need to calculate, right? So, can you see this place here? Okay, in this place, if you're talking about, hmm, just um, I am zooming in for you to have a better look into this. So, in this monohybrid cross, can you see the purple color? And can you see the white color flower? So in this flower, what we are doing is we are doing the controlled pollination. So when I'm using the scissor, when I'm using the scissor, okay, the for what for I need to use a scissor. Okay, you're seeing scissor in the diagram. Why I need to use a scissor? I need to remove the anther. How do you call that? Emasculation. How do you call that? Emasculation. For each and every process in the biology, there is a special technical term is there, right? So by using scissor, I am removing the anther. So what do you call that? Emasculation. Okay. I am transferring the pollen grain from one flower to other flower. And then I am wrapping the flower with polythene bag. Okay, that is called bagging. That is called bagging, right? So the moment when you are seeing the scissor, the moment when you are seeing the painter stick, so that means we are doing the control pollination. It is not happening naturally. Okay, so removal of the when I'm using the scissor, only one probability anther removal. What do you mean about anther? Male reproductive organ, or you will say male reproductive part. Male reproductive part, not organ. Anther is a male reproductive part which is having the n number of male gamete that I am removing in a bisexual flower. So why I need to do the anther removal? Why? Because I need to bring the control pollination. Okay. The anther removal is called what? Emasculation. Okay. But can you explain to me why I need to do this type of anther removal? Why? Because I need to bring varieties. What variety you're going to bring? If I am transferring the pollen grain from one particular plant to other particular plant, I'll be getting the chance for light purple color. I'll be getting the chance for getting the dark purple color. I'll be getting the chance for getting the dark. Okay, here we can say very good white. means we can see the very clear white. Okay, that color I can get. So all the probabilities we are understanding. So why we need to do in the bisexual flower? Why? Because in the bisexual flower, there is no chance for cell pollination. There is no chance of cross pollination. There is a maximum chance for cell pollination. What do you mean about cell pollination? The pollen grain is spreading the same flower. That is called cell pollination. The pollen grain is moving the same flower, cell pollination. Pollen grain moving from one flower to other flower, cross pollination. Right? So this is the way you need to understand. Right now, we are entering into the in the law of dominance. We discussed already the genes and alleles information for you. Okay, what do you mean about the alleles or nothing but gene copies? Like this, if you're writing, you will get only quarter mark instead of getting 1 mark. Then how I need to write the answer? Alleles or gene copies having a specific location in the chromosome. Okay, controlling an alternative pair of a character. Controlling an alternative pair of a character. Okay, like this, if you're writing, then only you'll get full marks, right? Okay, that is the reason why this class is exclusively conducted by Gotouniversity team. Okay, for making you to get maximum score in the board exam, right? So now we need to understand basically the stem height, flower position. These many number of varieties are there. Okay, we are observing the differences in the flower position. Okay, pod color, pod shape. Okay, can you give the example for the pod shape? Yes, one pod is round, one pod is wrinkle shape. Okay, one pod is having wrinkles, one pod is having a round shape. Okay, I got it. Right? Some seeds are round, some seeds are wrinkled. Okay, some flower are yellow, some flowers are green in color. Right? So depending upon the contrasting character, so we are choosing the plant and then we are doing the crossing for getting the better hybrid. Okay, in terms of disease resistance capacity. Why I need to do all these things? Because I need to serve all the population size with a very good amount of grains at a cheap price. That is why I'm doing all this type of experiment. Okay, and I'm getting benefited from this all these experiments, and that I am exploiting this information by giving many number of grains to the people who are living below poverty, right? For that only we are understanding all this type of crossing over, right? So now, if you're entering into the genotype and phenotype, each and every word is important. Any student can ask any question in the Q&A section. That is why what I'm going to the Q&A section and then I'm asking you students to pose the questions, right? So genotype and phenotypic ratio, when we are narrating, we are in the revision class. Okay, in this revision class, so even though some students are asking the basic questions, we are here to explain, right? What is genotype? So gene character narration, okay, in terms of ratio. One hybrid is having tallness, one hybrid is having dwarfness, one hybrid is semi-dwarf. I need to bring everything in terms of number, in terms of statistics. So which offspring came in the maximum number, that I need to narrate so that I'll understand which particular allele is dominant, which particular allele is recessive. Like this I can understand, right? So the mathematical expression, we need to understand, we know very well in the lower grade when we are doing the maths. So this equation is there. Okay, for reading this equation, we are not here. We need to bring what is hidden behind this formula. A plus B whole square is equal to A square plus 2AB plus B square. That we need to bring, right? So that if you're bringing this particular maths formula here, what we studied in the lower grade, then of course we can understand the concept of genotypic phenotypic ratio calculation in the case of monohybrid cross, right? So we got these many number of genotype or phenotype. So capital T, capital T, small t, and small t, small t. We got the three important genetic variety, but we got three important type of genetic variety, but we got two different type of tall. We got two different type of tall. Why it is called two different type of tall? This capital T is same capital T. That is homozygous. This capital T small t is what? This capital T small t is what? There is denoting the two different type of allele, right? So this capital T small t denotes two different type of allele. That means heterozygous. Capital T is homozygous. Capital T is homozygous. Capital T small t is homo. Okay, capital T is homozygous. Capital T small t is heterozygous. So the tall character contouring two different type of allele pair we understood by understanding the genotypic ratio. That genotypic ratio I got it because of the specific crossing over. How I did the specific crossing over? Why? Because I did the pollination by choosing a specific type of emasculation bagging procedure in bisexual flower. Yes, this is a way you need to understand by asking many questions in you. You need to ask questions. Okay, here opening the PPT and opening the book. Genotype represents the genetic composition. No, no. What is this formula speaks about? Okay, what is this homozygous denote? Same type of allele. What is this heterozygous speaks about? Different type of allele. Okay, can you explain what is allele? Gene copies. Okay, alleles are gene copies. Okay, occupying the same locus of the homologous chromosome, governing a pair of a contrasting character. This definition is not here. Okay, I'll close my eye and I'll say the answer, any answer as you like. So you should have preparation. I should have preparation. Then only the entire education system will develop. Okay, so saying allele as a gene copy will give less mark for you. Be very careful with this, right? So now we are entering into the next important test class. Is a type of backcross. Test class is a type of backcross. Test class is a type of backcross. Why I need to do the test cross? Why? Because I need to understand the gene composition of the parent. Okay. Then what is this test cross? A type of back cross. Okay. And what is the benefit of this? You are understanding the gene combination of the parent. Okay. Now the parent. Okay. Can you see this place here? And one is exactly the purple color, and one is white in color. When you're doing the crossing, what happens exactly in this crossing? Right. Test cross, a type of back cross is not mentioned in this slide, right? So, but we need to explain much more if the answer they are asking the 5 mark. So test cross, a type of back cross, which is specifically made to understand the parent genotype. Plus, we are doing the cross little bit different from the normal crossing. What do you mean about different cross? I'm not understanding. Okay, recessive homozygous. We are crossing with the F1 hybrid. Whatever the F1 hybrid you are getting, that the F1 hybrid you are crossing exclusively with the recessive parental type. Then obviously you will be understanding the parental information after getting the F2 generation. After getting the F2 generation. So, for example, if I'm crossing purple, selfing or self pollination. If I'm crossing the white and white, selfing. So selfing or self-pollination. So what I'm saying for you is, so the selfing, the symbol is represented always with the selfing, the symbol is always represented with, if I am rounding it, this is self-pollination or self-fertilization, or it is indicating selfing. So many terms they are using the board exam: selfing, self-pollination, self-fertilization. Okay, we are bringing it, okay, to bring the true breed. Why I need to bring the true breed? Why you don't want variation? Because certain type of plants, we, if you're taking about certain type of plants, if you're taking about the information, we need to understand basically the plants of various varieties or in a various geographical area. Some plants grows very faster, some plants grows very slowly, some plants grows very moderately, some plants grows moderately. Like this, what we are saying, many important varieties of plants, right? So what we need to understand basically is this is the geographical crossing over, specific crossing over. We are understanding with this. Now we are going for the genotypic composition and the phenotypic expression. In this particular, before going to this category, again back, I'm saying to you, any one of you are having any doubt in the case of Q&A section, that is why what we are asking you to put the question in the Q&A section. Why we put the Q&A section? Why? Because so we will understand exactly where you are exactly wants the question exactly, right? So can you see this place here? Test cross we explained. Now we are entering into the Punnett square. This is the Punnett square. So this Punnett square is also called as checkerboard. This Punnett, again back I'm repeating, this Punnett square is also called as checkerboard. In this Punnett square or checkerboard, the capital L and capital L, we are bringing it here, right? And then the capital L and small l we are bringing here, small l here, capital L, small l here, right? So from this information, what you are understanding? So here the capital L dominates the small l. Capital L dominates the small l. So what is this? The true breeding tall. Okay, true breeding dwarf, small l, small l. So either you give the capital L, small l, or either you bring the capital T, small t, anything as you like. Okay, so we will get this type of results very easily, right? So again, back I'm repeating, be very careful when you're drawing the Punnett square. So when you're drawing the Punnett square, see this symbol exclusively used for male. This symbol is exclusively used for the female. Okay, this symbol we are using exclusively for the female, right? So the above symbol is for male, the below symbol is for female, right? And then I told about the capital L denotes dominant allele and the small l denotes the recessive allele, right? So now we are coming for the next phase explanation is what do you mean about monohybrid cross? That we already told n number of time. If you are doing the crossing with the two different parent varying in only one character, monohybrid cross, right? So now we are coming for the law of dominance. In the law of dominance, what we need to understand basically is the law of dominance. In the law of dominance, what we need to understand basically, please keep your mind calmly and then understand the message. Any one of you can say what is in the law of dominance. Any one of you can say the example for the law of dominance. Any one of you can say the law of dominance information very, very carefully. Any one of you can say what is meant by the law of dominance. In the last session only we discussed with you. Any one of you can say law of dominance. So no one is saying this. Okay, okay, nobody is typing the messages in the case of law of dominance. It's okay. So dominant allele suppresses the expression. This is the thing I told to you. I given the time for you. Why? Because we are opening the Q&A section in the portals to understand if anybody posted the question. We are saying the answer at the maximum. Okay. Here, if you're talking about the in the law of dominance, again back I'm repeating, dominant allele suppresses the expression of the recessive allele. Nobody typed the answer. It's okay. But when you're asking the question, you should say the answer very faster. Heterozygous condition denotes two different allele determining the same character. The quite antonym of the heterozygous. Here, heterozygous is already there here. Okay, the quite antonym of the heterozygous is homozygous. Homozygous, antonym of heterozygous. Homozygous denotes two similar allele is responsible for. Two similar allele is mainly responsible for a particular character determination. That is homozygous. If two different allele determining a character, heterozygous condition. Like this, we need to understand. Okay, so Q&A section we are checking simultaneously. Okay, if any one of you can ask the question, please ask the question. I am there with you to help you. So now we are coming for the second phase explanation is before going to the law of segregation. Before going to the law of segregation, in the law of dominance itself, okay, I am going for the examples. In these examples, calmly you listen to me. Inheritance of the flower color. Inheritance of the flower color in Snapdragon. So in the CBSE board exam, instead of using the word Snapdragon, they'll say dog flower, 4 o'clock plant, mirabilis jalapon, antrinum majus. All the plants are having the same answer. That is the main advantage of this Gotouniversity class, right? So Snapdragon is also called as Snapdragon, antrinum majus, 4 o'clock plant, mirabilis jalapa, okay, dog flower. Like this, many examples we are seeing. Okay, all these things are showing are the examples for this, right? So now, if you're talking about the in the Snapdragon, the color of the flower is determined by a single gene with two allele, red and white. Red is dominant and white is recessive. Okay, the problem, what will be the genotype and phenotypic ratio of the offspring? Like this, they ask us to find out the answer. You can see the parent, okay, heterozygous red flower, okay, white flower snapdragon, right? We discussed this many number of messages, right? So in this, what we are understanding is the red should dominate the white, and we should supposed to get red, but we are not getting red. We are not getting red. We got purple. What do you mean about that? The capital R and small r brought the character together. That denotes the that denotes the that denotes the independent dominance. No. Incomplete dominance. Yes. What do you mean about incomplete dominance? The capital R brings off of its character, small r brings off of its character in a one particular progeny. So instead of getting red, we are getting purple color. Instead of getting red, we are getting purple color. That is a message we need to understand, right? So now we are coming for the next basic message. Again, back I'm repeating. Calmly listen to this concept. Okay. So here, what would expected a 1:1 ratio of the red flower to the white flower snapdragon plant in offspring? Okay, so 1:1 ratio. So here in the solution, this is the way you need to write parent 1, parent 2, set up the Punnett square, cross the allele. Okay, and then we are bringing the phenotypic ratio, genotypic ratio. So like this, all the answers are structurized with a lot of subtopics, so that what you will understand, you will understand means what the concerned paper evaluator will understand for getting the maximum score. This is the way you need to present the answer. Just writing a lot of big paragraphs will not give the maximum score, right? So now again back I'm repeating. This is a fantastic example for the incomplete dominance. It is not explaining law of dominance. It is explaining incomplete dominance. Right now we are coming for the next phase of explanation is nothing but in the inheritance of the seed shape in the pea plant. Okay, we know very well the seed shape is determined by a single gene with two allele, right? Round or and wrinkled, right? The problem is what will be the genotype and phenotypic ratio of the offspring? Okay, determining the genotype of the parent plant. Homozygous round-seeded pea plant, heterozygous round-seeded plant. Like these two different type of plants are we are crossing and we are getting the answer in this way, right? So the wrinkle seed plant in the offspring, okay, the round seed to wrinkle seed pea plant in the offspring. As also the offspring will have round seed due to the dominance of the capital or allele. This is the way the explanation or conclusion should supposed to be at the end of the answer. So denotes what? Conclusion, right? So this is the way you are explaining the answer when they are asking the question. These are the classic worked examples. So for example, if I need to score the maximum mark, what the Gotouniversity suggests. So this is the way your answer script should supposed to be, right? Now we are coming for the second phase explanation is tips for the exam. In the tips for the exam, here if you're talking about the practicing constructing Punnett square for the monohybrid cross, okay, these are the essential tool for predicting the genotype and of the ratio of the offspring. Punnett square construction or checkerboard construction that we already narrated for you in the concept where we explain in the concept the Punnett square. Can you see the presentation? Okay, this is the Punnett square. This is the Punnett square. Yes or no? This is the Punnett square. So this is the way you need to explain with the column. When you're explaining the column, then automatically what everything will come very nicely, right? Okay, your calculation will be very easy. Okay, the capital L, definitely dominant. Capital Li, definitely dominant. Small l, recessive, right? So that is the reason why in the case of the tips for the exam, we told to you for getting the maximum score, we need to draw the Punnett square. Punnett square, we already explained the content. And then in the Punnett square, what I told to you, we need to explain the male symbol. Just only narrated for you. This is the male symbol and the female symbol. We already given to you or not? Okay, both the symbols we need to explain in the Punnett square. Okay, and then second thing is, if you're talking about the Punnett square, determines the outcome of the process involving dominant and recessive trait. For example, predict the results when crossing two heterozygous individual. Okay, so here basically when the heterozygous individuals, when we are crossing, we'll get many number of offsprings. So we'll get many number of offsprings. Okay, the reason behind that is the many number of offspring. because two different alleles are participating in a crossing. That is the reason why what we are saying what heterozygous individual crossing. So solving monohybrid cross problem, dihybrid cross problem, uh, they will give calculations for you. So we calculate already 1:1, 1:3:1. Now only we calculated in the content. Okay, yes or no? Can you recall that? Okay, can you recall where we calculated? Okay, if you can't recall, I'll show exactly where is the ratio, right? Can you see this place here? We did the calculations or not? We did the calculations, yes or no? So this is the calculations we did, right? So where we okay always using the formulas and getting the calculated results and predicting with the proper explanation for the answer whatever you're getting for that particular numericals is very important, right? So we given the worked examples for you, okay, and then now we are coming for the tips for the exam. Okay, tips for the exam we discussed, right?